All right, welcome everyone. This is our third California Deep Ocean DDT community meeting. My name is Amalia Ruda Almada. I'm an extension specialist and researcher at the University of Southern California Sea Grant program. We have a full room, which is really exciting, and we have a large audience online via Zoom. So a few quick reminders. This meeting is being recorded. We have Spanish translation and captions available for our online audience. We welcome questions from the room and online via the Zoom Q&A function pictured here. We ask that you hold your questions until the moderator opens up the room during the Q&A sessions. And we welcome our online audience to add any technical Zoom questions to the chat at any time. So as we get started, we'd like to acknowledge the Indigenous peoples who for millennia have lived, thrived, and stewarded the lands and waters in this region. Under their reciprocal care, the ecosystems were healthy. We pay our respects to the Gabrielino Tongva, as well as the neighboring tribes, which are the Chumash, Tataviam, Serrano, Kawiye, Luiseño, Achimem, and Kumeyaay nations who still have deep kinship to the traditional homelands and all life therein. We'd also like to acknowledge the incredible teamwork behind the scenes to make this day happen, from our funders to the researchers to our planning committee and the small army of amazing people providing day of support. We thank you for your time and generosity towards this important cause. And a big thank you to all of you, our audience, for participating in today's meeting. Our goals for today are to make this an accessible and engaging forum to hear from funded deep ocean DDT researchers. And in addition to hearing about the research, to actually engaging and discussing as a diverse community here today. We have over 200 registrants representing agencies, researchers, community-based organizations, local industry, concerned community members, congressional representatives, tribal communities, and more. And with the diversity of attendees we have represented here today, we have a few requests from all of our participants. To be mindful and open to the diversity of perspectives and viewpoints represented here, to keep comments constructive to the meeting's objectives and respectful to community members, and finally, to keep in mind that the projects presented here are still in progress, so we encourage caution in interpreting the implications until the research is complete and peer-reviewed. Many of the research projects you'll hear about today are ongoing. Each new finding adds a new piece to the puzzle board. In some areas, we're starting to see a picture come together, and in other areas, the picture is less clear. And this is a scientific process at work. So we're thankful to the researchers for this opportunity to peek inside their process as this research is unfolding. And although not all of the research questions have been answered or puzzle pieces come together yet, our hope is that the information shared today provides transparency into what we are learning about the deep ocean DDT issue in real time. We also encourage you to check out a document we've put together with the help of the researchers regarding frequently asked questions about deep ocean DDT, what we know to date. The QR code is here, and we will also send it out to registrants after the meeting. Very quickly today, kind of how we foresee this agenda, we'll have some quick background about the deep ocean DDT issue in Southern California, followed by 3 research panels. Each panel will consist of a 15-minute talk, followed by 15 minutes of Q&A with all of our panelists up here. Moderators will open the room and Zoom for questions from the audience, as well as pull up questions that we got from all of you from your pre-registration. If we can't get to all the questions, we'll follow up. We've been really committed to doing that through our FAQ document and other documents. We'll then have about 15 minutes for updates from the DDT community. We'll close out with a brief 3-minute feedback survey to help us plan future events. We really pay attention to your feedback, so we really encourage that. For those in the room, we welcome you all to stick around for snacks and conversation. We'll have a social hour after we conclude at 3:30. And with that, I'll turn it over to my colleague, Dr. Leon Guo, to give context to why DDT is still relevant to our region today.
Thanks, Amalia, and hello, my name is Leon Guo. I am a research coordinator with California Sea Grant. So for those of you who may be less familiar, DDT is an insecticide that was widely used in the United States from the 1940s until its national ban in 1972. At its peak, DDT was considered a miracle insecticide because it was incredibly effective. But that same persistence of DDT in the environment and its unfortunate ability to accumulate in the food web is why DDT is still a concern today, over 50 years after it was banned. Southern California has a complicated history with DDT contamination. The largest producer of DDT in the United States, Montrose Chemical Corporation, operated in Los Angeles County from the late 1940s until the early 1980s. Waste from this plant was disposed through local sewers and ultimately discharged to wastewater outfalls on the Palos Verdes Shelf, which is shown in this map on the left. So during the 1990s, 17 square miles of the Palos Verdes Shelf was designated as an EPA Superfund site, and efforts to address the contamination there are ongoing. We now know that between the 1930s and 1970s, DDT and other chemical wastes were also transported via ships, such as the one shown here, to designated deep ocean disposal sites in the nearby San Pedro and Santa Monica basins. Often that waste was bulk dumped, not in containers, and this potentially led to a much more widespread contamination throughout these, throughout the basins. So this deep ocean DDT is the primary focus of our presentations today. The primary known deep ocean disposal sites for DDT waste are often called sites 1 and 2 in the Santa Monica and San Pedro basins, that are shown circled in yellow on the left-hand side. And as seen in this drawing of this very old depiction of the San Pedro basin seafloor on the right, these local ocean basins can be very deep and difficult to access, and at their deepest can reach just around 2,900 feet, or just over a half mile at their deepest. However, it's been a concern that the DDT contamination in the sediments at the bottom of these basins has potentially been affecting local marine ecosystems and human communities. So concerns around deep ocean DDT's potential to impact local ecosystems led to several important events in recent years. Beginning in 2022, California Sea Grant and USC Sea Grant co-led a deep ocean DDT research assessment workshop and subsequent report. In that, we brought together diverse groups to identify critical research needs around deep ocean DDT, including many of the researchers you're hearing from today. Around the same time, Scripps Institution of Oceanography and UC Santa Barbara were awarded multiple federal appropriations directed toward research of the impact of DDT disposal in the San Pedro and Santa Monica basins. And the state of California also was able to match the first federal appropriation of $5.6 million. And those funds are funding 4 research projects that are administered by Sea Grant on behalf of the State Water Resources Control Board in support of our understanding of human health risks and ecological risks in these deep ocean dump sites. So today you're going to be hearing from researchers who were funded by both of these sources of money. And we just want to note that people are very interested in the data that are coming from these very timely projects. And the agencies that are funding them are committed to releasing the data once the projects are completed through a peer review process. So luckily, these researchers are also presenting their results as it's ongoing, as is appropriate, such as in today's presentation. So in the last 2 community meetings, researchers shared important findings that have begun to eliminate the broad extent of DDT contamination in our local ecosystems. So researchers have found DDT compounds in sediments stretching nearly the entire basin seafloor, such as this image of the San Pedro Basin. The warmer colors along the seafloor indicate higher concentrations of DDT compounds. And you'll note that these go way beyond the yellow circle that indicates dump site number 2. They've also shown that this area has a long legacy of ocean dumping of chemical waste and garbage. This sonar map shows over 74,000 debris objects in this same seafloor area. So our researchers are continuing to work to better characterize where DDT is, how it's moving around, and whether it's been degrading over time. We've also heard from DDT researchers that DDT from deep ocean sediment could potentially be entering the marine food web. And recent research looking at deep ocean small fishes and zooplankton did find DDT in those organisms. So today we'll hear more from researchers whose work examines deep ocean DDT in other sea life, including in those that are eaten by humans locally. So just an important note that at this time, the EPA states that deep ocean DDT contamination does not pose a significant health risk to those who are engaging in local water recreational activities or to those who are consuming fish locally that are locally collected from this area, as long as they're following appropriate fish consumption guidelines. So this community meeting is really an opportunity to connect you with the researchers who are working to piece together the deep ocean DDT puzzle that Amalia shared earlier. So we will have 3 panels today. First, researchers will talk about where is the DDT, where it's been found, where it might be moving. The second panel will explore potential impacts of deep ocean DDT to organisms, all the way from those living in ocean sediments to coastal birds. And our final panel will focus on potential public health considerations, such as sportfish being consumed by local anglers and how microbes could be used to break down DDT. So with that, I will turn it over to our first moderator, McKenna. Sorry, do it the right way.
Hello, everyone. Thank you so much for being here. My name is Dr. McKenna Martin. I'm an outreach specialist for the Southern California Coastal Ocean Observing System, which is the organization that is tasked with managing the federally funded portion of the DDT research. I'm going to be moderating the first panel today, which you're going to hear about where is the DDT. Our lovely synthesis speaker is Dr. Sarah Gilley from UC San Diego and Scripps, and she'll be giving a 15-minute presentation that will be representing the research of 5 researchers here, including Dave Valentine from UC Santa Barbara, Will Barrelson, and Alex Sessions from the University of Southern California and the California Institute of Technology, Dr. Matthias Langhorst from UC San Diego, and Dr. Eric Terrell from UC San Diego. Without further ado, I'll turn it over to Sarah.
Thank you. So I'm going to talk about the detective work going into asking where DDT has ended up in this region off the coast of California. The talk I'm going to present is the work of the entire panel, and I'm going to go through this in 4 parts. First, asking where the DDT was originally dumped, then what happens to DDT once it's dumped, what was found at the dump sites, and finally, what is the present status of the DDT. So originally, and we just heard this in the introduction, DDT was dumped from the 1940s to the 1970s. There were thousands of tons of DDT dumped into the ocean in this region between Palos Verdes and Catalina Island. Really, this was done in 2 disposal sites. Much of it was nearshore dumping at the Palos Verdes shelf at a point, mostly at a point called White's Point Outfall. And then there was additional dumping as offshore dumping that Leon referred to, where the waste was bulk dumped. And it's estimated that on the order of 400 tons of DDT plus went into the water in the open ocean. So the bulk dumping is something we're still trying to understand, but and Leon showed these images of these barges that were loaded up with acid chemicals to be taken offshore. What we're learning as we explore this is that the dumping was more variable and more widely distributed than previously thought. That means that a lot of material was dumped. Some of it was radioactive waste. The dumping was not limited to just appearing on the shelf, and we continue to find DDT in new places, some of them surprisingly far from where we thought it should show up. So if you look through the records of the Freedom of Information Act and the documents, what you see is that this company, the California Salvage Company of San Pedro, California, had licenses to dump material, and I'm not going to read through all of these lines, but they went through and had permitting and licenses to take material, including radioactive material, and go through and take it offshore to dump it. There's documentation showing that it was in 55-gallon drums and barrels, and that it was taken in a variety of forms over time. So if we put stuff in the water, then the question is, what happens to it? And to look at this collectively as a group of researchers, we've taken a number of different strategies. My own research group has asked a simple question. Suppose we put particles into a numerical simulation and ask where they would go. So we have released particles, and if I can do this, you see this, in 2 locations, some of them at the Palos Verdes Peninsula, and we've let them get pushed around by ocean currents from a numerical model and ask where they end up. The shelf is relatively shallow, and so what it turns out is that over the course of a few weeks, these particles end up settling onto the sediment, and that's where they end up. They don't go further within our simulations. Then they're in relatively shallow water, and they could later on be resuspended or carried by turbidity currents into the deeper ocean. We also have tried putting particles into the middle of the San Pedro Basin at the point where bulk dumping might occur. And in that case, we see that they tend to fall, again, at a uniform rate. So you can see here, I'm not sure if I can show this, that particles are released and they fall basically due to gravity. They're relatively small, so they don't fall the way a rock would fall, but they fall. And within a month, we find that they've settled at the bottom at 900 meters depth, where then we face the question, what happens next? Can they be resuspended? And that's still an open question. It looks like it would take a lot of velocity to make them, a lot of ocean current to make them move again. So we are still exploring that. So then what's happening in this basin? This is a region here. You can see this is the basin we're looking at. Here's Catalina Island. Here's California. And the dump site 2 is right in the middle of this. There's a lot of ocean topography around here. There are sills and ridges. So there's a lot that can affect how sediment, DDT-laden sediment might move through this region. We have an expectation that at the bottom of the ocean, anywhere in the ocean, the currents should be fairly weak. But in this particular region, model studies show, and historic data measured from ships show that there is evidence for intermittent events with strong currents that might flush the water out of these deep basins and stir up the sediment. And so you can imagine a scenario then where particles might encounter a strong current that comes into the basin and maybe can resuspend it and move it further. And so that's something we're still trying to understand more about. One of the ways we're trying to understand this is by putting out a mooring, and Matthias Lankhorst and Uwe Send just last month put out a mooring in the San Pedro Basin at dump site 2 that has measurement capabilities, including bottle samplers intended to look exactly at what's happening in terms of bottom currents and a potential particle resuspension, as well as some passive samplers designed to pick up any chemicals that might be in the water that might stick to this passive sampler. So that's where the water goes. Then the question is, what happens to this chemical, DDT, once it sits on the seafloor? And so DDT is the parent compound, but over time it degrades into multiple derivative compounds that are referred to as DDE, DDD, or collectively as DDX, where X is the unknown. So this is not a simple degradation pathway. There seem to be multiple products that can emerge over time. What happens over time then depends on a variety of things. There are different daughter products that depend on the presence or absence of oxygen. And if we look at a sample water column, so here we're looking at concentration as a function of depth within the sediment of the seafloor. And what you see is that the very top of the sediment core, there's oxygen. And so that allows one set of chemical reactions. Below that, there's a suboxic layer with some metals, iron or manganese, that affect the reactions. And below that is a different set of chemical reactions. So each of those can lead to different degradation products from the DDT. Different subcompounds have different properties. And what is really interesting is that although DDT is relatively sticky and tends to stay attached to the sediment, some of these derivative products appear to be water soluble, and so they can appear in the pore waters of the sediment, and they can be potentially brought back into the ocean and transported with bottom currents. We also see some evidence from surveys in the region that there may be animals that live in the site and that can burrow or bring sediment back up to the surface. So that's shown here. You can see a worm here that is digging through the sediment and mounding things up and mixing the sediment within the water, within the sediment core below the water. So you can see images of that here. So there's a lot to understand about what that actually means for the availability or resuspension of potentially DDT-laden sediments in the water column. To try and understand that, we have funded researchers have been gone and really done extensive surveys to look at the seafloor. Some of that has happened through remotely operated vehicles and autonomous underwater vehicles that have surveyed the regions where dumping was done. So these maps show patterns of these surveys carried out by autonomous vehicles. I'll try and show this image here. This is an animation that shows the sorts of things we see on the seafloor. And what emerges from this is that there's an enormous number of objects on the seafloor, barrels, evidence for World War II munitions in various stages of corrosion or decay. Obviously, seawater is somewhat corrosive. Some of these may have had caustic chemicals in them. And so it's not clear how stable they are after several decades sitting on the seafloor. The other aspect is to try and understand what's in the sediment. And so there's been extensive surveying done to look at sediment cores across the seafloor. A typical sediment core looks something like this. So it has sediment over the top few centimeters, and somewhere several centimeters below the surface is a layer that is full of DDT and DDT derivative products. And then below that is a sediment layer that does not have DDT. What that suggests is that sediment was laid down over a period of time during maximum dumping, and it has subsequently been buried. When you look at what's in the sediment, what you see is a very broad range of products. And I show that in a larger image here. So these are pie diagrams, show different DDT and DDT derivative products. And what you see is they have really different distributions in different locations. So that implies that the chemistry of the water or the distribution was not completely homogeneous throughout the region. And so if we try and put that together into an image of how this has evolved over time, this is showing DDT, DDD, and DDE evolving over time, mapped from these sediment cores. And what you see, and I'll probably have to show this again, is that DDT on the left is initially not so high, and so that's in the deepest sediment. Let's see if we can play that again. And then over time, you see an infusion of DDT and the derivative products evolving over time, so that DDE on the far right ends up persisting longer than the DDT and DDD. Wait for that to finish. Okay, so in the present day, then you're probably asking how much DDT is left, how much DDT is degraded, and how much of it is toxic. And the short answer is that we don't totally know that. The good news is that a lot of the DDT has been buried under ocean sediment, so it's potentially a little bit sheltered from the ocean circulation. But the bad news is that this is an evolving story. The processes that move DDT and move sediment are still being explored. There are new daughter products still being discovered. And some of these products are water-soluble compounds that have been detected in the sediment, and that means they move out of the sediment and back into the water. And the more we look, the more we find. There's new sampling locations, in particular to the west of Catalina, that have us thinking we don't fully understand even what was put down or how it got there. So what's next for researchers and consumers? Well, obviously we like to talk about remediation, but we can't really find a full remediation solution until we understand the basics of the issue, the scope of the dumping, the locations, the types of compounds, and their toxicity. So we're working on answers to all these questions. What actions should be taken? Well, there are potential for regulations on ocean activities that could at least avoid disturbing bottom and sediments with DDT. So for example, we might imagine a no troweling requirement to try and avoid disturbing this sediment that has been successfully buried at least under a couple centimeters of non-DDT filled sediment. And more information is coming up in panels 2 and 3. Panel 2 will address potential impacts for sea life, and panel 3, the public health considerations. So at this point, the other panelists and I are happy to take questions about any of these issues. Thank you.
Thank you so much, Sarah. I'm going to open up the floor to our panelists. Does anyone have a question for our panelists? We have 2 online questions.
Maz Dajiri asks, if DDT wasn't produced until the 1940s, where did the DDT come from in your 1935 slide?
Yeah, so that, I guess that was my slide, so I'll take that. Yeah, so those were the sediment strata of those age. And so there is a little bit of turbation that happens. There's animals that live within the seabed that move small amounts of DDT up and down. So that 1935 strata, there was clearly no deposition directly into that strata, a small amount coming in years later when things were moving it down into that layer.
Thank you, Dave Valentine.
So yeah, Dave Valentine, UCSB.
Any other follow-ups, or should we move to the next question?
We have another online asking, what is the cause of the different composition of daughter products for DDT? Thinking of the pie graph slide.
Is that Will Barrelson?
Yeah, Will Barrelson, USC. The degradation of DDT is complicated, but there are steps of progression that have to do with the gaining of electrons or losing of electrons. I'm putting it in a very loose term, but it has to do with redox, the amount of oxygen available to drive some of the reactions, the degradation reactions. So you can imagine that that degradation could be occurring in an organism that ingests some DDT, and then inside the body, the redox conditions may be different depending on where that DDT resides and how long it spends there. Or the products could be formed in the environment. If it's sitting on the surface of the sediment, it might be exposed to oxygen. If it's buried within the sediment, it may not have access to oxygen. And so the degradation of these compounds, in simple terms, is related to the redox environment, or the presence and absence of the oxidation reduction potential of the environment. It has a lot to do with the presence or absence of oxygen.
Can I just add one additional component onto that? When DDT, the parent product is transformed. It can happen to some extent chemically, but mainly we think it's happening by microorganisms. And so what we're seeing are the preferences of the microorganisms that live in these different niches within this environment. And that's why we see the buildup of some things, then not buildup of others.
Question at the back.
Yeah, I'm just wondering, you've been looking at this for a while. Are you finding any sort of, so loud? Hi, Mark. Any sort of spatial boundary at this point of where you're actually finding DDT contamination of significance? And also, have there been any hot spots that have sort of popped up with the supplemental work since last we've heard from you?
Okay, that was Mark Gold. Casey, I think you forgot to mention his name, Dave Valentine again. I think that one's going to me. So we have been trying to map. We've taken samples now at 129 different stations. We've been expanding out. We have seen some hot spots. There's a hot spot that's west of Catalina that has us looking more closely, that's in a bit shallower water. It's one sample so far, so we really want to get out there and make some confirmations about that. We've been mapping to the north. We're well up into Santa Monica Basin, and we're still seeing relatively high concentrations, meaning things over 1,000 micrograms per kilogram in the sediment, sort of halfway up into Santa Monica Basin. So we're trying to find the boundary there. When we look in Santa Barbara Basin, it seems very low. So somewhere between Santa Barbara Basin and the middle of Santa Monica Basin, I'm guessing there'll be a steep drop off. And there seems to be a fairly steep drop off in concentration as you go to the south. So we think we've hit the edge. In the southern part of San Pedro Basin, there seems to be less accumulation that happened down there. So that's all to say, the northern boundary we're working our way in on. There's some stuff going on to the west that we don't understand. In the south, we think we have reasonably well nailed down. There's a second part to that question. I have no recollection of what it was. So.
You actually hit it at the beginning, which was hot. You named an intriguing hotspot, but I don't know if there were other ones. And so what would you say now the DDT field is from the standpoint of acreage?
I'm sorry, can you say that again?
Acreage, what's your estimate right now on how big the DDT field is?
I forgot to do my polygon math before I came, so I'm going to keep my mouth shut.
In the middle here.
I had a follow-up question, I think, for Will, and that's with the pie diagrams. They show a lot of heterogeneity, but are they all from the same quote unquote stratigraphic layer, like the oxic, the suboxic, and the anoxic? I mean, hopefully the pie diagrams aren't mixed.
Okay, so Will handed the microphone back to me because his team made those measurements, and Alex Sessions and Jim at Caltech, I think, did the measurements on Will's project, but they were samples that came out of our archive, so I think I have a better handle on where they came from. They were not from the same depth. Those were from different depths. So part of the variability that you're seeing is because we chose some things that were in that DDT peak and some that were maybe above it. But it still doesn't track one-to-one, right? It's there, yeah. So that's part of it, though.
Yeah, and also, without getting into so much detail, we're on the right side of this map, you see the two pies that are right next to each other, sort of side by side right next to each other. That would be the pie on the left was more towards the surface of the sediment, and the pie on the right was deeper in the sediment. So that's a case where we're showing shallower and deeper. And you see that the deeper one has more of the red color. The red color is the DDT, the parent. But there's these unusual and somewhat very distinctive distributions of these compounds.
In the very back.
Has there been other dumping sites up and down California or in other states, and has the issue been dealt with differently at those other locations?
Much of our work is focused on this particular area based on earlier reports of where items were dumped. And I'm familiar with a 1970s report from SCCOOS that lists, I believe, 14 dump sites total. And of those 14, 1 and 2 stand out for chemical dumping. So at the others, there's also a lot of stuff dumped, but chemical waste is not called out. And I think that is the reason why we're focusing on 1 and 2. Correct me if I'm wrong. But
No, I think that's accurate. The only thing I wanted to add to that is that we certainly know about other dump sites that are in other locations outside of Southern California. So this is not just a Southern California problem. There are dump sites off of every major coast from the U.S. So the differences are what was dumped and our level of knowledge. We're learning about these dump sites. We really haven't learned much about most of those other dump sites. So this is a bit of the canary in the coal mine in terms of how do you even go about studying a dump site. And I don't even exactly accept the idea of a spot on the map as a site, right? This was all spread out. There wasn't like a location that somebody actually went to, right? There's a spot on a map, but that was kind of irrelevant and really put there afterward in a lot of ways. So I think of it more as dump areas. And there are a lot of those both off California and elsewhere.
But also, because I'm the oldest one in the room, there was a period of very intense oil drilling in Southern California, and the water's offshore, and we know that. And the drilling for oil involved pumping down of water that's heavy, and you don't make water heavy naturally. You have to add barium to the water to make that water stay down in the drill hole. So all this barium-rich water, barium coming from barite, is a mineral. That barite-rich water, all these particles of barite in it, was dumped. And that barite settled in these So part of the dumping grounds were designated because of oil waste deposition, as well as the problem that we're discussing, which is the chemicals in the DDT.
The question to that.
Can you talk a little bit about the composition of the actual DDT waste product? I know they had a caustic and an acid waste. How much of which metabolite was in the waste product?
Yeah, I think we don't have an exact answer to that. We know a bit about what was in it. So the recipe to make DDT was strong sulfuric acid, and that's most of what was really dumped. Monochlorobenzene as a primary reactant, and chloral, which is a 2-carbon unit with 3 chlorines and some oxygens, as the third main ingredient. So we expect that there's each of those in the waste. Maybe excess of monochlorobenzene is likely. So think of the waste as probably mainly sulfuric acid, some monochlorobenzene, and then whatever DDT didn't precipitate out in the precipitation reactions. So you have this big reactor and you're trying to crystallize. It's actually more of a waxy globular material that forms, and then you separate that out, and you have all this acid waste that then you have to deal with. So that's sort of the bulk composition of what we think was there. And then there's whatever byproducts, whatever was in the monochlorobenzene, because they weren't using really pure grade chemicals. So we have strong evidence that there were things like trischlorophenylmethane that ended up in this waste stream. We're still trying to figure out exactly how much, but it's a concern. As well as potentially some of these other compounds that are breakdown products may also have been in the waste stream, as well as different isomers of DDT. So we don't really know what the ratios of those things were. We're honing in on some of that with the research we're doing, but it's a tough answer if you're not a chemist.
In the middle.
I had a question, I think, for Will again, and that's in the actual cord sediments. Is there any evidence for these strong intermittent currents? Like, are the sediments coarser than you normally would think, or is there any microstratigraphy? Anything that the geologic past would indicate these currents were active?
That sounds like a question from a geologist to a geologist. Short answer is no. There's There are zones, places where we've looked and we know very well that there are turbidites, that are deposits, coarser grained material that originates on the shelf and the slope and flows down into the basin. We know about those and we can map those out. But in the normal course of sedimentation in San Pedro Basin, there's really no change in sediment type.
Great, well, that's all the time that we have left for questions, unless you had one last thought. But thank you guys so much to our panelists, and thank you to the audience for the questions. And we're gonna bring up our second panel now, the impacts to sea life.
Hello, good afternoon, everyone. Thank you for joining. Our next panel is What are the potential impacts to sea life? My name is Tanya Torres, and I am a marine debris research associate with California Sea Grant. And I'm going to be moderating this panel with Devin, who will introduce herself.
Hi, everyone. My name is Devin Rossi. I'm a California State Sea Grant Fellow with the Fish and Game Commission, and I'll help Tanya co-moderate this panel, and I'll pass it back to Tanya.
Thank you. Our synthesis keynote speaker today is Professor Nathan Daughter, who is a research scientist and adjunct professor with the School of Public Health in San Diego State University. And then after his 15-minute presentation, we will be joined by Dr. Charlotte Seed, who is a museum scientist with the Scripps Institution of Oceanography, as well as Dr. Unha Ho, who's a professor in the Division of Environmental Health at San Diego State University as well. So with that, I'll pass it over to Nathan.
Thanks. Good afternoon, people. I'm going to be presenting work on, the last presentation I had about sediment, and this is going to be about what happens to those chemicals that are in sediment, how and in what way do they enter in to the food webs, and what do they do there. And this work is, it's representing funded research from at San Diego State University and UCSD SIO. I have to figure out how to use this. Okay. So what I'm going to talk about first is what we knew before this work was funded. I'm talking both the federally funded and the state funded projects. So our group at SDSU had been through other funding sources working on generally looking at contaminants in the upper trophic levels. And in the course of that work, we identified a lot of DDT and DDT-related compounds. We found that DDT plus is very complex. And so what I'm talking about here is I'm going to use the term just DDT, which is what I think conventionally for decades people measured. DDT, that term refers to both of a specific chemical, but also a group of about 6 to 8 closely related structures. And what we found in these marine mammals, sea lions, dolphins, condors, were many structurally related compounds similar to DDT plus. So these are just two of the major forms here. If you don't know how to read the chemical structures, just think of it, they're slightly different shapes, right? But what we found in the upper trophic levels was many more forms, something maybe 45 more different chemicals. Some of those are still undefined. We know that they have this sort of backbone, you could call it. So they're probably coming from the same source, but we haven't identified the chlorine positions quite yet. These earlier studies, the distribution, we know that they're there, but they weren't quantified in those studies very well. So we don't really currently have a good idea of the amount and distribution in the sea life quantitatively. And that's what the current funded work is trying to determine. And this is important, but because these chemical structures relate exactly to the toxicology and then the risk of these contaminants. So, and also what we knew in the start from a very large body of work is that DDT does bioaccumulate and biomagnify. This is from regions around the world. DDT is one of the class of what's called persistent organic pollutants that are regulated for the most part worldwide. And there's a large amount of models and field studies on this accumulation and bioaccumulation, biomagnification of DDT. That biomagnification is going, what that means is that we're bioaccumulating from the sediment, and more precisely the pore water that surrounds the sediment. That's sort of the first transfer step into the organisms, usually in the sediment. That's the biomagnification. I'm sorry, that's the bioaccumulation. And then biomagnification is going up the food web. Basically, these chemicals are retained in the fat, the lipid. And so when one animal eats the other, if it's not excreted, it just accumulates in the next animal. And you get the highest levels, you know, on a wet weight or lipid weight basis. The highest levels occur at the highest portion of the food web. So that's presumably where the most biological risk might be. This can't really be predicted very well, the biomagnification. Some parameters can be predicted from the structure, but things like metabolism, you can't really predict. We have to do these measurements. So the question is, we knew all of this about DDT, maybe not specifically to the food web of the bite, but just sort of generally, we know that these processes happen for DDT, but we don't really know about DDT plus yet. That's the other thing we wanted to look at. And then we knew both generally, but also still recently, that there are adverse biological impacts from DDT. Generally, DDT, and specifically its metabolite DDE, it interferes with the formation of the eggshell. And then that caused low reproduction rates, you know, early on, decades ago, in a series of seabirds in the area. This was well known. But even just recently, I mean, in the past, within 5 years, studies have shown that California condors that feed close to the coast and have a higher DDE load, they have a lower fertility due to eggshell thinning. And then a completely different toxicity pathway is that California sea lions, the cancer is caused by this herpes virus, but there's this association, probably due to something like a weakened immune system, that shows a link between the cancer and the persistent organic pollutant level, including DDTs. So what did we want to assess? Given that background knowledge, what did we want to assess for this situation here? And there was four areas. So one, how does the DDT plus chemical profile change? Is it bioaccumulates and biomagnifies? This is pretty similar to the question that was presented in the last panel for the sediment. So if this, you know, we're going to go through the trophic levels here, the food web, and if each of these boxes is a different individual form of DDT plus, it might start out in one way in the sediment, and of course, as we first discussed, there's also a puzzle here, even what's happening in the sediment. But as it goes and biomagnifies up, we might see something very different in marine mammals, for example, than we do in invertebrates. What makes this problem difficult is even as I just mentioned, and it was mentioned in the last panel, the abundance and spatial distribution in the sediment is still an ongoing question. And then again, this cannot really be predicted. We have to measure this because all the different forms of DDT plus are going to have a different bioaccumulation and biomagnification potential. The other thing we want to assess through this work, it's going to be done for the sediment too, is this chemical profile that we see, this whole range of DDT plus, is that unique to this region? We know from our previous work that DDT, not DDT plus, but DDT levels in our regional marine mammals are among the highest in the world compared to other studies. We know that DDT plus is present in these marine mammals and at the upper level of the food web, but we haven't rigorously assessed those concentrations yet. Can this DDT profile be attributed to the waste dumping? That's basically the question we're looking at. And just to highlight, again, the bounds of the contamination is not really known yet, as was mentioned, and even the two dump sites is probably, we're learning, is not a good idea to even talk specifically about those two dump sites that have been on older maps, as we're looking at some much larger area of contamination. Because this is a production waste issue, this is going to be different from the DDT that most other studies have looked at, which is an agricultural runoff issue. So that's the final sold product. That's the pesticide that's applied to farmland that runs off as stormwater or something like that, versus dumping of the actual synthetic chemical waste. Those are two very different things. And that probably means that this DDT plus is unique worldwide. Or maybe, again, DDT is not banned everywhere. There's some areas that still produce it. We might have something to learn there too. Okay, and then another thing we wanted to look at was, what is the DDT plus body burden of important and/or-sensitive species? So there's kind of two ways to look at it. Some species might have sensitivity toxicity-wise, like, for example, the eggshell singing. So we want to get a handle on what's going on there. But even if a species is not necessarily affected in terms of the toxicity, it still may be a very important, what you might call, vector for transfer to other animals that are highly impacted as it goes through the food web. We want to understand both of those two issues. This is complicated because it's the food web in the Southern California Bight. The Bight is a very productive area, so the food web is very complex. And also we have different types of species movement. So we have right now, I guess you could say, undefined area. The bounds of the sediment contamination aren't known. We're going to have some resident species might overlap with that, so maybe we don't know they're bound either. And then we also have migratory species that are coming in and out that their prey is the resident species, for example. So it gets kind of complicated. These boundaries, of course, are hypothetical there. And then our ultimate goal, in conjunction with the toxicity, is to assess the risk. So just to explain, so we have a chemical hazard, which you could think of as the toxicity of the chemical itself. And then we have an exposure level, right? So you need both of those things to assess the risk. So if there's no exposure, for example, it doesn't matter if the chemical's toxic or not. And then there's sometimes contributing factors. I mentioned the sea lion cancer. The herpesvirus is a contributing factor that needs to be present to initiate the risk for those chemicals. So our funded projects here, I'll go through next. Again, it's been constructed so that we're looking at these three different levels. So invertebrates, lower food web, upper food web. And I'll just say that for the status, the samples for all the projects I'm talking about have been acquired. And they're currently in different stages of being analyzed. So I just have a little bit of actual data to show. Okay, I wanted to start off with just the acknowledgements for the SIU CSD side, just to highlight it's quite difficult to get the samples. So we just wanted to acknowledge the captains and crews of the research vessels. Okay, the first project for the invertebrates is to really understand what invertebrates are there. This is, again, the critical step from the sediment, sort of the first step of the bioaccumulation, and then right before the biomagnification takes place. And I'll say they're going to use this, the DNA to identify these organisms, some of which are known, some of which are new to science. So those are being cataloged, and then the DDT plus is being measured right now. The next project is now looking at what we're going to call the lower food webs. The questions are, do these deep food webs around the offshore dump sites accumulate DDT plus? And then can we determine, or does this help lend evidence to that the DDT plus came and originated from the offshore dump sites? There's a little bit of this work has been published here, and I'm going to highlight just one of the several findings from that. But one of the things that was done was the same species of fish were collected pretty much right over the so-called dump site 2. And then 100 kilometers away in the Santa Cruz Basin, the same species of fish were sampled. And what was found is that those two contain pretty much the same DDT plus contaminants and the same relative amount. Probably that's not, I hope I don't say something wrong, but probably the zone of sediment contamination doesn't extend that far out. But what it means, the potential spray species are migrating quite far away. Okay, now I'll talk about the upper trophic level next, and this is the acknowledgement slide for that work. And there's a large number of very important collaborators that acquired the samples for the work I'm about to discuss. I wish I had, the data to show for this, but instead I'll just go through the different, it's all about the upper food web, the different aspects that we're going to look at for this. One section is on both the sea lions. So one is on the sea lion pups. So the aim of this study is to look at the association of DDT plus with different maternal foraging areas. So there's two groups. One forages over the moms forage over the zone of impacted sediment there. Mm-hmm. And then the others are on the upper Catalina Islands. Those mothers mostly forage away and then to the north. So we're going to look and see if the DDT plus levels in these two groups are different. And then second, we're doing sort of the phase 2 of the sea lion cancer study. So I mentioned that earlier. That was all done on DDT only. Now we're going to look at the sea lions with DDT plus and a few other changes too, to look at that association between cancer and DDT plus. Next, we'll also look at, we have 2 bird studies. One is on birds, 3 groups that have different diving depths. We're going to look at bird livers. And then we're also going to look at California condors. So condors, they have, the reintroduced condor has a coastal or inland habitat. Those that have a coastal habitat, they're going to eat, the dead stranded carcasses. They are thought to have a higher DDT load, and that contributes to the eggshell thinning. We're going to investigate that for DDT plus. And finally, the temporal trend, it's sort of surprising how much interest there was in DDT. There's a Superfund site related to it. But once the problem was considered solved back then, the work mostly stopped. So there's huge, I would say, pretty big gaps in the resolved temporal trend information. So we wanted to take a look back to that. So we're using archive samples in both cases. One is with the short-beaked common dolphin, which through other work we determined is a very good sentinel species generally for contaminants in this region. And then second, sort of tied with the previous study I mentioned, as designed to also look at the temporal trend in these archived condor blood samples. Okay, and finally, the next panel, we'll talk about this more, but we're also looking carefully at the toxicology. Just to kind of briefly say, there's 2 types of toxicology tests being done. One is cell assays. You could think of that as more molecular level specific investigation. And then we're doing whole animal toxicity, which looks at more of a broad range of possible mechanisms of toxicity. Okay, and then the last slide is we're working with David and Gail, a writer and an illustrator on they wrote a children's book that we're involved with. And I was trying to think of some way to conclude this. So as a rule, I'm kind of trying to summarize what the book is trying to say. So as a rule, we could consider that all major pollution events, no matter of what type, they should be investigated, right? That new scientific understanding can be applied to other current or future similar events. In our case, we want to make sure that there wasn't an important biological impact that was missed. But also we need to, and I think this is what the book is about, we need to look at the public awareness and trust in our processes. Is the pollution contained, and does the major pollution go down once controls are enacted? Even if we can't do anything about it, I think the public would want to know that. And finally, storytelling like this, it can be used to promote public respect and care for the environment. Right? Thank you.
Thank you, Nathan. I'd like to invite our other research panelists to the front.
All right, great. Thank you so much for that presentation, Nate. And I understand that many of the project results are still in progress. So just to kick our discussion off with a broad question, how can understanding the impacts of DDT on sea life inform better management decisions such as remediation, risk assessments, and or broader ecosystem level effects? Just kind of putting it in a zoomed out perspective before we get into more specific questions.
Okay. That's a really hard question, but I can give one. It's hard to talk about broadly, but there's one specific story I can talk about. So this is the California condor. So it has the coastal and inland, the two habitats that they're introduced in. And so the coastal one, it feeds off of the marine mammals. So it has a high DDT load, and then it has eggshell thinning. And it does have lower reproduction currently. So that's a known problem. And so as they're looking, but overall, the program is fairly successful, right? Most people for the condor population is getting larger. So as they're looking for new places, there's a large number of factors that are involved. I mean, condors also experience most of the toxicity actually comes from the lead shot that they could eat. So that's sort of the biggest problem. But the DDE is a secondary factor. So as they're looking for new places, we worked on a study with the San Diego Zoo that they might be able to reintroduce the condor more in the Gulf of California, so on the east side of Baja California. And they can, using the pro-offered food to the condor, they can kind of train the condors to go certain directions. So we looked at the marine mammals from the Gulf of California, and they have a lot less contaminants than those off of Southern California. Does that make sense?
Yeah, that's helpful.
So that's an actual, and again, the management decision is a lot of different factors, logistical, lead shot, there's a whole bunch of things, but the DDT contamination would be one of several things that they could look at when they make the decision about where to make new condor reintroduction sites. That's one type, but that's sort of a unique situation. Obviously, sea lions and dolphins are not reintroduced in certain locations like that.
No, that's really helpful, Nate. That's an interesting regional case study that we have here off the California coast, and appreciate the explanation to that very hard question. I can open it up to the floor if we have any folks online that have questions that they've entered into the Zoom chat.
Yes, we have one. Even if the current risk is low because some of the chemicals are below the sediment, isn't there a serious risk due to the fact that they could be released in the future? In other words, can we just depend on DDT, et cetera, being buried as we consider risk?
When we talk about the risk, it's very difficult, so Paul, the panel 3 can talk about it, but I think there's still very unknown factors here, so, the panel 1 also talked about a lot of degradation products, but in our panel 2, and then we're also talking about this diversity of the DDT plus. So a lot of data that we depend on is that those known 6 DDT compounds, even that 6 DDT compound, there's still gap, in terms of the toxicity. But then if you think about like a Nate showed, like a multiple DDT plus chemicals, we don't know that their toxicity, their biological effects, or their ecotoxicity effect, so it's very difficult to say, what's the risk at this point. But, we hope that we can define the hazard. This is what we're talking about, hazard, and then we're talking about exposure, and then the risk, like the risk to humans or the risk wildlife species. So it's quite complicated. Hope that we can answer some more questions, Yeah.
I recall panel 1 mentioning a specific form of DDT that is more soluble than previously expected. So even if we do nothing, if the animals do nothing, then chemistry is going to take its course. So that was humbling, but also very important to learn.
Sorry, just one. So it does seem both the sediment and the sea life and the toxicology, we do need a lot more time simply to study it before we start to think about what can be done about it, because we don't even know what it is yet exactly.
Do we have any more questions online, or should we jump to folks in the room?
Yes, we have one from Alan Mearns. Hi, Alan. Are you looking at mesopelagic fish, historical contamination of myctophids, McGregor versus now?
Yes, we do. That we actually looked at the actually the Leonis group and looking at the temporal trend of the McToffid of the DDX, and also in our lab that we are analyzing them for more larger suite of the DDT, you know, that we talk about this DDT plus compounds. Yep.
Should we jump to you in the middle? Do you want to run the mic? Thanks so much.
I can easily imagine marine mammals going down a few hundred meters and eating things off the bottom, perhaps. But do they go that deep? And or what goes down to 900 meters and eats things off the bottom there? How does DDT at 900 meters in the food chain on the bottom of the ocean end up back up to things that live at the top, near the top of the ocean? Who's moving it up?
So I'm not a biologist, but I learned it, you know, from collaborating this study with the the SIO scientists, researchers. The middle trophic level of the food web is extremely important because those small fish, you know, the small organisms, they're living in the deep ocean, but they can also go up all the way to the surface. So if you think about 900 meters, they're actually living in the very deep ocean, but they also go up to the surface as well. So this is why I think that this study is so important to, like, in our previous studies, that we found the highest DDT concentrations in marine mammals, but in there where they came from, you know, and then this middle trophic levels organisms are very important, you know, to tell us, you know, that deep down sediments could be the source of that, you know, the top trophic level organisms to DDT. And then I'm talking DDT plus.
I think that's one of the most interesting and maddening things, that there is no direct connection. I'm not aware of any individual species that dives all the way down. So how is it making that mysterious journey? And in some ways, maybe the sediment is a bit of a red herring if it's also coming from other spots. We talked about dump regions and diffusion. So yes, I don't think there's an easy answer.
Any other questions?
Yeah, I had a question, and maybe for Nate. Thinking about the invertebrate burrowers, they should be a special target because they're the one creature that really ingests that sediment. And they seem to be the most likely, you know, to uptake the DDT plus compounds. Are you able to capture these burrowing invertebrates with dredge samples or any of the sampling that we take, and are they seen as a special target? And the last thing, I don't know how important they are in the food chain, but they seem like the creatures that would most uptake chemicals.
Sure, I can take that from the invertebrate side. So we also thought along those lines, but I do want to emphasize, first, all of them are absolutely tiny. So I was showing you photos that were taken under a microscope, but the largest of them would be like the size of ants. And one burrower that particularly caught our interest, I was on a cruise with Dave Valentine, and we saw some tubes of parchment worms, Phyllochaetopterus limicolis. They're maybe the size of the diameter of thick spaghetti. So again, we're talking very small, and they're vertically oriented. So we thought maybe they're acting like straws, or here's something that's directly connecting the surface where all the oxygen is to maybe the relevant layers of DDT. So with that in mind, the Levin lab at Scripps is doing DDX analysis. Because these creatures are so tiny, it's a huge pain to amass enough material to do these analyses properly. You need grams, and again, we're talking about fragmented spaghetti. So with enough patience and sampling, again, in these really sparse regions, we were able to cobble together enough material, and the Levin lab is running these. So maybe that worm would be the one. My suspicion actually is because it's vertical, it's only intersecting that DDT like rich layer in a very small surface area. So, and the worms themselves stick their palps up at the surface. Everyone wants oxygen. So the animals are physically touching the bottom layers of the sediment, but where they're feeding is where the oxygen is at the surface where the DDT isn't. So I could see it going either way. Great line of reasoning. We'll see what the data say.
Can take another question in the back here.
Thank you, Mark Gold. A two-parter. The first one is more direct, which is just to get some overview on, since I think we saw 8 or so studies that were described, on what your relative ballpark is on when you think you'll have some results to share, would be one thing. And then the second one, which I know I've talked to Una a lot about over the years, is that have you made any progress on fingerprinting and really seeing, you know, just to remind everyone in the audience, we have DDT hotspot in San Pedro Bay at Dominguez Channel. We have another one off PV Shelf, and then we have the deep water one. You know, PCBs in the first 2. We don't really have PCBs in the second. Now we're obviously, you know, the degree of chemistry and what we know now versus what we knew 30 years ago is night and day. So what are you finding out so far on the fingerprinting side and really getting a better idea of where the contamination's coming and the organisms you're looking at?
Yes, Mark. So the first question, when we're gonna get the data, you know, so this funded study, it's really focusing on quantitation of the DDT plus. So we actually established the quantitation method, you know, pretty good right now. And then we are assessing the samples are crazy, you know, right now. So hopefully we can get the data probably sometime next year, definitely, you know, so that's what we're expecting. The second is the fingerprinting analysis. You know, we also made a aim in this study, a subset of the each species that Nate showed, that we wanted to do not only the DDT plus, but we wanted to put all kind of halogenated organic compounds, which including PCBs and other chlorinated, brominated chemical contaminants in this species. So, so we have some of the samples, the data actually was very interesting. You know, that, for example, like a new species, like a brown pelicans, you know, and we find a lot more other chemicals as well, additional DDT related chemicals, you know, and there's something surprising chemicals that we did not expect, you know, so we hope that we get those things, like we already have some data like dolphins and sea lions, condors. We hopefully get those fingerprinting. Currently we got the brown pelican, and then also the trophic, mid-trophic organisms, you know, we published the data, you know, those organisms, we also did the same non-target analysis. So we got those fingerprinting data for that. And then we also have like a macrophage, and so we have a lot of this fingerprinting, what we call it non-target analysis. We sort of like can get the ideas, you know, what these chemicals are actually transport and spread, you know, among the food web, and also hopefully geographically distribute the data. But it's pretty challenging, but it's actually very, very exciting.
And it looks like we're at time, so I just want to give a huge round of applause to our panelists who have to distill all of this incredible research into a digestible presentation, and thank you to our audience for thoughtful questions. We appreciate you.
We'll now transition to panel 3.
Hi, good afternoon, everyone. My name is Dr. Catherine Pease. I'll be moderating panel 3, where we're going to be talking about the health considerations of DDT. So I'm excited to introduce Professor Carrie Sant. She is from San Diego State University, as well as more recently, Michigan State University. So she'll be talking about projects from a number of researchers who will be joining us for the panel afterwards, and I'll introduce them very briefly now. So she'll also be talking about the projects from Dr. Johanna Gudleben, who is from UC San Diego, Scripps Institute of Oceanography, Dr. Katie Lyons from the Georgia Aquarium, and Dr. Brendan Talwar from UC San Diego, Scripps Institution of Oceanography. All right, thanks. Welcome, Carrie.
All right, try to clip this where I'm not too loud for everybody. Usually a problem. All right. All right, so thank you all for being here today. Talking about the public health considerations for DDT is a little bit of a loaded question, right? As you've seen from these projects, we haven't done a lot of human biomonitoring, but there's a lot of ways that we can infer things about public health from other information. So again, we just introduced all of our speakers. So my perspective on this presentation today will be to introduce the fine work that's being done by all of these individuals so that hopefully we can have a really rich Q&A session following our presentation. The way that we can start to address some of these things about human health, despite not having human health data from this region, is through what's called the One Health paradigm. So One Health is a concept that's used globally, mostly for biomonitoring. So it's an integrated, unifying approach that aims to sustainably balance and optimize the health of people, animals, and ecosystems, right? It recognizes that the health of humans, domestic, and wild animals, plants, and the wider environment are closely linked and interdependent. So we just had a great presentation from Dr. Dotter, right, who was discussing about sea lions and also having increased risk of breast cancer, right? Does that mean that a human might be more prone to develop breast cancer if they're also exposed, right? So we use things like One Health to help inform that because there's some processes that are conserved between animals and humans that we can learn a lot about. I'm going to talk about that a little later in this presentation. So again, we've been hearing about this all day so far, right? What is DDT's health legacy? What do we know about it? So when I think of DDT, I'm a toxicologist. When I was first learning about my field, we started with DDT. It's classic, right? It's a pesticide. It was a really good pesticide because it's incredibly neurotoxic, right? And those same mechanisms that make a fly susceptible to DDT, we also have that in our brains and in our bodies, right? Now, the difference is if I spray DDT on a fly, that fly is really, really tiny, right? So ultimately, that fly is getting a higher dose of DDT than a human might get, right? We might get the same amount of DDT, but honestly, we can kind of get rid of it pretty quickly, okay? So really, as a toxicologist, it would be very misstepping if I didn't mention the dose makes the poison. That's the paradigm of my entire field. The amount that you're exposed to ultimately translates into a health outcome. So with DDT, one of the most classic things that we've seen over time are reproductive and developmental toxicity endpoints. And in particular, there's this concept called endocrine disruption. And again, all that fancy terminology, it messes with hormones. All of us know what hormones are, but it messes with hormones. So one of the classic ways that we look at DDT is actually through modification of estrogen or androgen signaling. So when we talk about DDT, a lot of people see it behaving kind of like estrogen. So when we're looking at the effects of that, who knows, what do we think, what do we associate estrogen with? Shout out from the environment. Well, women, typically, right? We associate it with reproduction, we associate it with pregnancy, we associate it with menopause, right? Those are all things that, imagine if you're being exposed to a chemical that mimics estrogen. That's going to play a toll on your body. And likewise, there's other compounds and famous studies of other pesticides and fungicides, et cetera, that have endocrine disruption, where imagine you're exposing males to that compound, right? Now that's an exogenous source of estrogen that could affect their hormones and their signaling in their body. But ultimately, that's what leads to the things like eggshell sagging in birds, reduced or perhaps overstimulation in early puberty, reduced fertility, and increased pregnancy loss at really high concentrations. But also we associate it with carcinogenesis. So IARC, who's the international group, whenever we talk about like class 1, class 2 carcinogen, the international organization that makes that determination is called IARC. And IARC has labeled DDT as a class 2A carcinogen, meaning that there's high evidence in animals that it's carcinogenic. And from that, we infer that it's a probable human carcinogen, especially for breast and liver cancers in particular. And part of the reason we see it higher in breast and liver cancers is because those are really fatty tissues. DDT is really lipophilic. It loves to go in really fat-soluble bodies, right? So it goes into things like adipose tissue, it goes into things like the liver, because that's where the fat in your body collects. So I have the absolute privilege today of giving a brief introduction of some of the projects in hope that you will all have really good questions for each of these panelists in a moment. So I'm going to start off first with presenting the work of Dr. Joanna Gudelbun and then also Doug Sweeney, who is not on our panel today, but did some of the work that I'm going to be presenting. And their first question was, how does DDT plus, or DDX in this case, impact microbial communities? And how do microbes influence DDX degradation and bioavailability? So really briefly, again, I'm hoping you guys are asking them the right questions. They're the experts, not me, on their projects. We've been talking about increases up the food web, but let's start at the smallest level, which are microbial communities, which are super important, right? As we keep discussing and we just addressed in the last panel. So microbes are the base of the food web, and they're the main nutrient recyclers, right? So if we disrupt the microbes that are in an environment because of the exposure, that can have cascades going all the way up through the food chain, right? So in their project, one of the first things that they did was look at what types of DDX are found in those areas, but additionally, how is that influencing the localized microbial communities? So one of the things Joanna can be questioned about in a moment is looking at the difference, in particular that dump site 2's microbial community is different from the other locations, and that DDX concentrations might not be high enough to exert detectable selective pressure, influencing the total microbial communities on those areas. And other things like oxygen and nutrient availability are also contributing to those dynamics. And likewise, there we go, okay. And also they did mesocosm studies, right? So think of this as setting up, just like it's shown right there, little bottles, right? We are adding DDT into what we're going to call a local, just small ecosystem, a snapshot. It's how we create an environment in the lab, right? So if we put DDT into those bottles, what was interesting is that they found that there was conversion or metabolism of DDT to those other metabolites, right? So what they were looking at was basically the process by which we degrade DDT into those other metabolites. So when we talked about DDX being found in all these different animals, et cetera, that's already starting to happen at the microbial level. So part of what even those lowest organisms are being exposed to might be actually the metabolites, not the primary compound. And that also the oxygenation, so if you look at abiotic conditions, those more highly influenced that, right? So in the abiotic one, let's see, do I have a pointer? Great. So if you look at this side, those are environments that have higher or less oxygenation in that particular point, right? So if you look, they're actually more likely to be metabolized. So next, question 2. How does DDT plus or DDX move through food webs? And that's Brendan's project. So the goal of their research was to look at how much DD plus is in commercially and recreationally important fishes of the Southern California bight. Ultimately, what this is really getting at is, okay, we know that some of this is getting into fish. Is it fish species that we're eating? Are we likely to be exposed? So Dr. Dotter mentioned the issue of risk, right? If we know that there's hazard, right? If we know that there might be toxicity data, are people going to be exposed? So looking at historical samples, they found overall that if you look on kind of this x-axis orientation here, if you look at the amount of DDT in sediment, it's very strongly correlated with DDX in fish. So what we can take away from that is that if there's higher DDT concentrations in the sediment, you're going to have DDT concentrations in those fish that are higher, right? And if you look lower in the actual cascades, you'll find that benthic ones even have a stronger association. And lastly, also, this is looking at a temporal study. So as we move along here, you can find that this is 1990, 2000, and 2010, right? And this is the total DDX that's being found in lanternfishes in particular. And DDX or DDT plus concentrations in those lanternfishes have been declining over the last 30 years since DDT was banned. So next I'm gonna highlight the work of Katie Lyons looking at are DDT plus found in commercially relevant fishes. Katie gets to work with cool species. I'm really jealous about that. But we have, in this particular case, she's presenting her work on the bat ray versus the angel shark. What's interesting is that even though these are two very distinct species, they have different ranges that they go over. So in particular, bat rays have a wider habitat versus angel sharks tend to stay local. So what Katie and her associates have found is that DDX concentrations are highest in angel sharks around Los Angeles. Now remember, those are ones that stay around Los Angeles, right? So if we know that there's dump sites here, this makes sense. We expect to see higher concentrations of DDX in those areas and in those fish that have stayed locally their entire lives. And lastly, I'm gonna talk about the work that we've been doing. So what happens once exposed? So I talked about the issue of endocrine disruption, right? And I talked to you about estrogen. Well, do these things actually have estrogen activity? We have decades of information showing DDT does. Cool, but what about, as Dr. Ho and Dr. Dotter covered, the other 45 compounds, right? Like, what are they doing? So started off, and this is a project with the San Diego Zoo. We looked at, there's two estrogen receptors, alpha and beta, which is a fun fact, but in humans, but also in California condors who have similar estrogen receptors. And what we found is that most of these DDX have estrogenic activity that are early metabolites, and that they have stronger estrogen activity actually in humans than condors. So we've been talking about eggshell thinning and some of these reproductive effects in a condor, but actually if this is estrogen receptor mediated, you're more likely to have a stronger effect in humans than we would in condors. That might be leading to some reproductive effect. And then lastly, I want to say, are these things even similar? So again, we only really have toxicity data. I'm a toxicologist. It's embarrassing in that sense, but do we have toxicity data for these things? Not really, right? We have it for the key ones. So DDT, DDE, and DDD, we have pretty robust toxicological information. But again, if we're talking about 45 different compounds, well, a lot of them just don't have any, right? So what we've started doing is I work with a fish model called the zebrafish, and we use that as a model for fish, no surprise there. But also they have 70% gene homology with humans. So if I want to study effects on estrogen, if I want to study effects on the brain, I can do those studies in a zebrafish, and it's actually fairly comparable to what we would expect to see in a human. So we exposed zebrafish embryos, so think of it as during pregnancy, to a bunch of these other DDT plus compounds. And what we found is that certain species of the DDT plus behave similarly, and they have similar conformations of their chlorine positions, whereas a lot of the metabolites in particular behave very differently. And from the previous presentations, including in panel 1, that might be due to different exposures from different sediment areas, et cetera. So your exposure profile or some of that fingerprinting that we were talking about might actually influence the toxicity that's happening in your body, right? So we're working on that. So stay tuned. This is fairly new data. With that, I want to say in summary, we know DDX concentrations may have affected microbial community composition, and there's evidence that microbes can metabolize or degrade DDT. DDX can bioaccumulate and potentially biomagnify, and it's correlated with sediment DDX concentrations. DDX are found in commercially and recreationally important fishes, so there is room for human exposure through our food web. And lastly, DDT plus have endocrine activity, which suggests they may potentially have impacts on reproductive and developmental physiology, just like we know for DDT. And with that, I want to turn it over to our panel.
All right, great. Thank you so much, Carrie. So we'll go ahead and open it up to questions, I think, in the room. Is that okay? Yeah, go ahead. Yeah.
Okay, Dave Valentine, ECSB. So in some of the data that we saw from the first talk, DDOH, sorry, this is for you, Kari. Everybody's looking at me very anxiously. Some data from the first talk, we saw that DDOH was in many samples the most abundant of the DDT plus. And on your last plot, I noticed it was the lowest curve. Does that mean we have the least to worry about? Or can you expand about, should we be worried about DDOH given its abundance?
What a great question, Dave. In short, I can't answer that question completely right now. We're still working on it. But does it mean that we shouldn't care? No, it doesn't. In that last plot I showed, the exposures were all the same concentration. So this is taking into account mixtures. And one of the hallmarks in toxicity is, okay, well, some of those things are behaving pretty similarly. If we have them all in a mixture together, do we just add it up, the effect that it's having on the body? Does it multiply the effect when we have more of those things together? So I wouldn't actually want to say that DDOH wouldn't have an effect for that reason.
All right, thank you. Any other questions? I see one in the back there.
Good afternoon. My name is Albert Ramirez. I'm the president of ILWU, Local 56. We're Hazmat Maritime Union. This is critical information. We would love this information shared with our folks in the community in San Pedro. We actually drove out here, some of my sisters and brothers. We actually work in the maritime, and we do remediation, oil. We've been following this story, great reporting by LA Times. And this is critical because our rank and file are exposed to this. And it's iridescent of like the DDT with the farm workers. And now it's in the marina. And I think there was a gentleman that talked about the Dominguez Channel and Pedro. So we drove out here to get this information to share it with folks in the community in Pedro, Wilmington, and even Long Beach. But we want this over there in our neck of the woods. So thank you for this information, because this is very, I wouldn't say alarming. We knew about it, but this is something that I think a lot of our folks in the community would appreciate. So kudos to everyone for making this possible, but let us know how we can help, because we're out there in the ocean, so to speak, with our mariners and our hazmat workers. So thank you.
Great. Yeah, thank you for attending. Appreciate it. Yeah, do we have some online questions?
Yes. We had one about, is anyone studying what vulnerable populations are eating? Say they're worried about subsistence fishing communities.
Yeah, Brendan, do you want to take that?
Yeah, our lab, Tony Slew's a PhD candidate in the Simmons lab at Scripps, and she's going to be working with a local nonprofit called Heal the Bay to analyze historical peer angler survey data in particular, and also supplement that with contemporary surveys to try and get a better understanding of who's eating what and who's aware of these surveys, and how we might better reach the people who aren't aware of them in the future.
Just to follow up for our project, so our species that we chose, and there's a third one up there that we have data forthcoming on, but taking it from an elasmobranch perspective, so our cartilaginous fishes, right? We know a lot of sport fish, tuna's really sexy, but you need a lot of money to go on a party boat to go fish those animals. And we have people that are going out to the pier for free catching directly from these waters. So our project was actually taking that social justice component, and that's why we focused on bat ray and leopard shark as those things that are easy for someone to go and get, and angel shark as our commercial cheaper protein, right? So it's cheaper than swordfish, it's cheaper than those other sexy fish. But those are important for us to look at because those are what people are eating, and they might not have access to understand these things for whatever reason. And so that was a big component of those species that we're targeting.
Great, thank you. Yeah, question over here.
Hi, so I believe when you were talking about how DDT moves through food webs, you mentioned that you've seen a decline in the past 30 years of the concentration. And I was wondering how that relates to the commercially relevant species like the white croaker that are found on those subsistence, the shore communities by subsistence anglers.
Yeah, we've been analyzing the lanternfish from the Kalkofe long-term collection. So far, we have data going back to the 90s, but there are samples going back to the 1960s, and we're waiting for those. We're about a third of the way through right now. And the data are suggesting from that time series, as well as other papers that have come out, that these concentrations are going down in fish tissue across species in the Southern California Bight. So yeah, it's nice that things are lining up.
Okay, I have a question, and it's with brown pelican populations, and apparently the DDT affected their shell strength, and the populations dipped during DDT times. They seem to have recovered. I wonder if they fully recovered to pre-DDT levels, and if they have, is that a proxy, for the effect of DDT on higher organisms? Yeah, that's a question.
What a great question. I don't know if I have an answer for that for you. Nate, do you know? I know I'm probably not on the panel.
Maybe panel two.
I think, again, I'm not a marine biologist, I'm a chemist, but my impression from the sea lions is that there's a lot of other factors having to do with ocean upwelling and their access to prey and everything that probably control their population more. But the sea lions are affected, or there's an association between DDT exposure and the cancer prevalence that, again, is initiated by the virus. So I don't think total population is a proxy because there's too many other factors involved. And also it's important, I think, even if population goes up, the human population goes up, but we still study disease in humans. You know what I mean? I think the ecology is sort of the same. We want there to be a healthy ecosystem regardless.
Let's go to Dave here.
Yeah, Dave Valentine, UCSB. A couple of the previous responses made me think about some of the more recalcitrant components of DDT plus, in particular, the compound that we know is TCPM, which is chlorophenylmethane. What I'm wondering is that we heard statements that over time the concentration is going down, but we didn't hear exactly the concentration of what. And what I'm wondering really is whether or not there's any data to say what's happening with things like TCPM that are more recalcitrant. We maybe don't expect to be going down in the same way, if that's been looked at all, and sort of how we parse that statement of going down.
Our collaborators at SDSU are looking not only at DDT, but also a number of other compounds, including those. So we will have data to answer that question in the next year or so.
Let's go to Chris and then Bill.
Yeah, so it's for many of these top predators, marine mammals, birds, sharks, we've seen populations recover. In fact, they were recovering faster in the 90s when DDT was probably having impacts, but yet populations have come back. So the question is, as these degradation products are shifting and we're getting some of these potentially more toxic products, that really should be the focus, because we don't see necessarily population effects from some of the earlier products. So the question is, are those going to be more toxic, and if so, in what way?
Yes, no, that's exactly where my mind is going with all of this. Especially, you know, for estrogen activity, some of those ones that had the highest estrogen activity aren't routinely monitored, right? So if we are talking about that, yeah, we might not be necessarily seeing it in the population. Kind of like Nate was just saying, actually, just because there's more animals recovering doesn't mean they're healthy too. But then maybe it's just those more subtle effects, right? Even though it might not lead to mortality, et cetera, that doesn't mean that they're doing well. So that's kind of what we're getting at with some of these activity and behavioral assays, is just finding out like what's going on and do we need to be more concerned about some of these metabolites? Because for TCPM-OH in particular, it's quite toxic when we've tested it so far. So, and that's not routinely monitored. It's kind of been off the radar for a long time. So yeah, I 100% think yes would be the answer.
We'll go to Will and then an online question, and then I think we might have to wrap up.
All right, thanks. So Johanna, I say let's call the bacteria the first responders. They're in the mix, and you showed data that indicated, I thought, that bacteria don't really care if they're in a layer of DDT or not. The same population and lots of DDT or little DDT. But then you showed these jar experiments where clearly bacteria are doing something with DDT. They're degrading it, whereas the absence of bacteria, the jar has a different. So they're capable of, and in the jar they do, but in the nature, we think probably they don't. Is it because the jar, all they have to eat is DDT?
So in the jars, we did add an external carbon source.
I'll eat spinach if that's all there is.
We added, what was it called? Pot mix. We thought somewhat likely carbon source that is somewhat recalcitrant, so it doesn't get all used up at once, just to give them an additional stimulation of some external carbon source as well, because there have been other experiments similarly where they really tried to have only DDT as a carbon source, and there nothing has happened. There has been no change in the other things to eat. There has to be other things to eat because just the DDT, exactly, that just DDT itself is too recalcitrant as far as we know now to just be degraded as is, or the microbes that are able to actually metabolize it are in a very low relative abundances. They also need some stimulation to just like grow in numbers so that they are actually able to degrade it. So what we're finding in the mesocosmos right now, but this is again very much work in progress, and we're still like looking at how to analyze that data properly and to get like all the repeated measurements. So what we are seeing now is that the DDT is degraded to DDE, but currently it kind of stops there. So that is that sufficient? Is that a degradation? Maybe it makes it worse as some toxicity assays are showing. So that, yeah, I'll get back to you about that in like a year or two.
All right, do we have time for?
Quick comment from Maz Dagieri from LA Sanitation. He says, the city of Los Angeles has a rig fishing program that collects sports fish. We conduct fish tissue analyses, including DDT and breakdown products. We also have sediment chemistry data and outer LA harbor fish tissue data. LA County Sanitation Districts has its own data from the PV shelf. So just some resources to consider. And a question with this from Mary, with this newly identified hotspot to the west of Catalina, how volatile is this to the island community and visitors? There are a lot of commercial fishermen here, and we routinely eat halibut, swordfish, sea bass, etc. How concerned should we be?
Yes. Oh man, I mean, I'm just gonna say the same thing I said earlier. It's not like I'm gonna stop eating fish tacos, right? Not gonna happen. So I do think that there's something to be said again for the principle of risk. So just because we know these things can be toxic and we know people are exposed doesn't mean that there's actually risk of these major health effects, right? I presented on carcinogenesis. Does that mean you're gonna get cancer if you're eating this few times over the limit? No, right? It doesn't necessarily mean that.
So I do think that that's important for context. But in terms of what we can do, I mean, I do think that additional communication about fish advisories and things like that is important. But really, until we have all of this additional information, I don't necessarily know how to answer that question.
I would just add that for now, you can look up Good Catch California through the Office of Environmental Health Hazard Assessment. And that's where all the state advisories can be found, both at the regional level and across the state and nearshore waters if there isn't a regional advisory. So there's a lot of good information there.
All right, well, let's join me in thanking panel 3 and
Okay, so we have about 10 minutes. This is our portion for kind of general community updates. So we have one update from Sea Grant. Valerie Kuo has been an amazing intern this semester working on DDT. A number of different projects, that FAQ document that we QR coded, a video project about Will Barrelson's work, and she's going to talk to us a little bit about an ongoing project to get kind of the research updates online in a visually appealing and easy to navigate way. So I'll turn it over to Valerie.
Thank you so much, Malia. I'm going to preview buttons. Okay. So this summer I started with creating a list of questions to state-funded researchers doing research on DDT to kind of gauge what they're working on and what sort of findings they've been discovering, as well as what they find are the big upcoming research needs in DDT. So in developing this website, we wanted to have a more public-facing display of what is going on within ongoing research. I first started with talking to Professor Barrelson with talking about what sort of research he's doing at USC on DDT through an interview, which is also featured on this page. And we want to expand this website to also not only state-funded researchers, but also federally funded ones. And so some of the pictures that you'll see here from the website as I go through are stills from the video that I took, and as well as some photographs that were taken during the January cruises to collect some pore waters for their experimenting from Matthew, who is working as a research assistant. So I'm just gonna scroll through and just display some of the work that I've gotten. And we really wanted to make sure that the language on this website was really accessible to people. I know a lot of the things that we've heard today are really technical. And so making sure that anyone who comes across this website is able to have these really technical terms defined in a way where they are able to start from scratch. So here's just an overview of what the project consists of, and then some really great images again. We really wanted to make sure that the objectives were divided up and really clear of what this project is trying to accomplish. And then as well as some of the findings, we broke them down through each objective, making sure that we have some of the approaches defined over here. And then in looking at the methods, we wanted to show some of the different tools that were used, for example, the blades. And these images also show some of the coring that happened. And so by having more visual elements, we wanted to make sure that people are able to understand such a complex research that Barrelson is doing here. And then this is like a 5-minute video that I was with the help with a lot of the Wrigley interns as well, with their feedback. So yeah. And then we also wanted to make sure that there was a way for people to stay in touch with USC Sea Grants.
Thank you, Valerie, for all your hard work. So Valerie is with us through the end of the semester. So Will's page is going to go live first, but then she'll be following up with everyone else. She's been working behind the scenes to have this launched, hopefully by early 2025. We have some time for, we're going to open up the floor in a minute, but we did have John Chestnut from EPA who wanted to share some brief remarks based on the last few panels. Hopefully we can get him live. Yeah. Okay, John, can you raise your hand? Your virtual hand.
Yes, where? Yeah, it's not working.
In the meantime, if there's any updates you'd like to share, I know there's many of us in the audience working on DDT in different ways. So we just want to hear if anyone has a 1 to 2 minute update. Be thinking about those while we get John set up. Or we can come back to John. That's easier. Okay, John, I think you're good. Go.
Okay, I think I've been unmuted. Good afternoon. I was actually going to invite Dr. Renee Jordan-Ward from my staff to speak to the issue of efforts EPA has been working on with regard to trying to affect behavior around seafood consumption for the last 20 years at Palos Verdes Shelf. Is it possible to unmute Ms. Ward?
So they'll just have to raise their hand as well.
Hi, everyone. Are you able to hear me?
Yes, loud and clear.
Great. Thanks so much. This has been a really amazing opportunity to hear about the ongoing research. And I just wanted to point out the EPA does lead a large public education outreach program that aims to minimize risk to human health, particularly through the fish that are contaminated with DDT and PCBs from the Palos Verdes Shelf. This is a collaborative effort with other federal and state agencies, as well as local organizations. And we have several programs that we try to educate the public about fish contamination from PV shelf and the potential health risks of eating fish that are contaminated with DDT. Part of our outreach program includes active outreach to anglers. So we are out on the piers actively engaging with anglers at 9 piers off the coast of LA every single week, from Santa Monica to Seal Beach. And then we also have active outreach at local bait shops where we provide educational materials about DDT contamination from the Palos Verdes Shelf and health risks for consuming contaminated fish. And then we also group, I don't know if you guys have heard of that, the Fish Contamination Education Collaborative. That's our group of federal, state, and local organizations. But produce outreach materials that we hope are getting out to the public to make them aware of fish contaminated with DDT and PCBs. And we also attend local events, particularly around areas that we know anglers are coming from. And we're committed to outreach specifically to subsistence anglers to make sure that they have the information to make informed decisions about the fish that they are consuming.
Great. Thank you very much, Renee and John. We appreciate you sharing that information, and we can distribute that after the meeting as well. Anyone want to have the stage for a remark or two about what they might be up here around DDT. We're also happy to distribute materials after the meeting.
Can I ask a question for Renee? Sure. Yeah.
Renee, are you still there? Maybe not. Maybe we can do that offline, if that's okay. Yeah.
Are you able to hear me?
Oh, okay, she's back.
Sorry.
I just had a quick question for Renee. Thank you for that update. With the fish monitoring and also just the education outreach, how often does the monitoring happen? And when was the last update on just the fish advisory?
So we do not create fish advisories. We follow the fish advisories that are set by the state, particularly OEHA. And so we our program is to really educate the public about fish contamination and in particular a focus on these that may have a negative impact on their health.
All right, thank you so much for sharing those updates. Before we conclude, we do want to note we have some QR codes up here. We're asking everyone to please help us ensure that we can improve future community meetings. We will have at least one more in the next year that's funded by the state-funded awards. And we also have this Deep Ocean DDT listserv, which is where we usually announce events such as these. So make sure you're signed up for that if you want to be part of this in the future. I also want to give a huge thank you, especially to USC Sea Grant as our physical hosts, and Amalia for being really like the lead organizer on getting this event together. If you enjoyed it, it is really all thanks to her, the USC Sea Grant team, and then all of the collaborators, partners who helped bring this together today. And thank you also to our researchers, our synthesis speakers. It was a lot of work to get us here today, and I think they did a really amazing job. So, like, can we please put a hand together for them?
Yeah, I'll just say thank you so much. And Leon undersold how much she contributed as well. So I want to say thank you to the California Sea Grant team. And again, to our planning committee, Heal the Bay, SCCOOS, and Sacred Places Institute. We're really thankful for the collaboration. Thank you again for being here, coming to USC campus. It's amazing to see people in person. What a difference that makes. So we appreciate you contributing to the day in that way. All right, we've got snacks. Stick around. Our hope is that this is a time you can actually meet some researchers and ask them questions. No grilling, but polite if you ask them questions. And if you have any other questions about the day or what Sea Grant is doing around these DDT community meetings, please come to the Sea Grant teams. Thank you so much. Well.